Welcome back to a quick training video on cardiac action potential. First, let's dig into the cardiac cell. Cardiac cells, also known as cardiomyocytes, are the specialized cells that make up the heart muscle. They have unique structures that allow the heart to contract rhythmically and pump blood effectively. Here are the key components of a cardiac cell. Cell membrane, sarcolemma. Similar to other cells, the cell membrane or sarcolemma in muscle cells surrounds the cardiomyocyte, separating its interior from the extracellular environment. It regulates the passage of ions and nutrients in and out of the cell. Myofibrils. Cardiac cells contain myofibrils, which are cylindrical structures composed of contractile proteins called actin and myosin. These myofibrils are organized into repeating units called sarcomeres, which are the fundamental units of muscle contraction. Sarcomeres. Sarcomeres are the functional units of muscle contraction and consist of actin and myosin filaments arranged in a specific pattern. The interaction between these filaments during contraction generates the force necessary for muscle contraction. T-tubules or transverse tubules. T-tubules are invaginations of the cell membrane that penetrate into the interior of the cardiac cell. They help transmit action potentials deep into the cell, ensuring synchronous contraction of the entire cell. Sarcoplasmic reticulum, SR. The sarcoplasmic reticulum is a specialized type of endoplasmic reticulum found in muscle cells. It stores and releases calcium ions, which play a crucial role in triggering muscle contractions. Calcium release from the SR initiates the interaction between actin and myosin, leading to muscle contraction. Intercalated discs. These are specialized structures that connect adjacent cardiac cells. Intercalated discs contain gap junctions, which allow for the rapid spread of electrical impulses between cells. They also contain desmosomes, which provide mechanical strength and prevent cells from pulling apart during contraction. Nucleus. The nucleus of the cardiac cell contains genetic material, DNA, and regulates the cell's activities, including protein synthesis and cell function. Cardiac cells are highly organized and interconnected to form the myocardium, the heart's muscular layer. The coordinated contraction of these cells, initiated by electrical signals, allows the heart to pump blood effectively, maintaining circulation throughout the body. But what about their functions? Let's look into cardiac action potential. The cardiac action potential differs from the action potential in neurons. Cardiac cells have a specific pattern of depolarization and repolarization that is essential for the heart's function. There are several phases in the cardiac action potential. Phase 0. Rapid depolarization. This phase involves the rapid influx of sodium ions through voltage-gated sodium channels. This influx of positive charge causes the cell membrane to depolarize rapidly, initiating the contraction of the heart muscle. Phase 1. Initial repolarization. Following the rapid depolarization, there's a brief initial repolarization due to the transient opening of potassium channels, allowing some potassium ions to leave the cell. Phase 2. Plateau phase. Unlike neurons, cardiac cells have a plateau phase where the membrane potential remains relatively stable. This phase is primarily sustained by the influx of calcium ions balanced by some outward potassium flow. Calcium ions entering the cell help in muscle contraction by triggering the release of more calcium ions from the sarcoplasmic reticulum. Phase 3. Rapid repolarization. In this phase, voltage-gated potassium channels open further, allowing a large efflux of potassium ions out of the cell. This causes the membrane potential to rapidly repolarize back to its resting state. Phase 4. Resting potential. The cell returns to its resting membrane potential, ready for the next action potential. Next, we will look at the sodium-potassium pump for each action potential. Sodium-potassium pump in Phase 4. Resting state. The pump utilizes ATP, adenosine triphosphate, the cell's energy currency, to transport ions. It is an active transport mechanism. At rest, the sodium-potassium pump actively transports 3 sodium ions, Na+, out of the cell and 2 potassium ions, K+, into the cell against their respective concentration gradients. The pump continuously works to restore the ion gradients after an action potential, contributing to the repolarization phase by extruding excess sodium ions and bringing in potassium ions. It helps reset ion concentrations during phases of the action potential, contributing to repolarization and ensuring the proper sequence of ion movements during contraction and relaxation. In summary, the sodium-potassium pump plays a fundamental role in establishing and maintaining the ion concentration gradients necessary for the generation and propagation of action potentials in both neurons and cardiac cells, contributing to normal cellular function and electrical signaling in these cells. Thank you for watching our videos. Please like, share, and subscribe when you get a chance.